Well, good evening, everybody, and welcome to another episode of GUcast. This is Declan Murphy, broadcasting from not Peter Mac this evening, Renu.
No.
Where are we?
It's amazing. We're at the Sofitel.
We're at the Sofitel, Collins Street in Melbourne, which is not our normal abode, is it?
No. Are you actually here? Aren't you on sabbatical?
Yes, I am on sabbatical. Thank you for asking. But tonight I'm on sabbatical at the Sofitel with you.
Yeah, it's fantastic. It's a great milestone tonight, isn't it?
Yeah, it is exciting because why are we at the Sofitel in Melbourne?
This is the eve of the inaugural ProsTIC Preceptorship. 2022.
It is ProsTIC, which is what? What's ProsTIC again? I can't remember.
Oh gosh, you're testing me. How many glasses of wine have you had? It's prostate cancer, it's theranostics, it's imaging, it's a centre of excellence.
I think you got them all the ProsTIC letters.
Yeah.
Fantastic, well done. Yeah, it's fantastic. So it's the eve of the inaugural ProsTIC Preceptorship, which is this full-on, very immersive conference focusing on imaging and theranostics in prostate cancer, led by the ProsTIC team at Peter Mac. So it's exciting times. We've got a couple of great days ahead of us, haven't we?
Fantastic. I mean, it really is a culmination of such incredible collaboration between urologists, nuclear medicine physicians, radiologists, medical oncologists. It's fantastic.
It is. But I have been on sabbatical. Thank you for
You
asking.
have, yeah. So welcome back.
Thank you.
Are you back?
No. So thank you. Sabbatical is going to be 6 months of this year, which I'm very excited
Declan,
about.
you're killing me.
And I'm in the start of my first 3 months of sabbatical, and my last 3 months, and my sabbatical is focused on, drum roll please.
Yes, I can see you going through the change.
Yes. It's not focusing on more surgery. It's not focusing on, anything else to do with diagnosing prostate cancer. It's to do with nuclear medicine. That's right. I'm doing a sabbatical on nuclear medicine, and it's fantastic. My good buddy and collaborator Michael Hofman, who is a friend of the podcast, said to me, Declan, you seem to like nuclear medicine. Why don't you take some sabbatical time when it comes up and Actually, I think he was saying learn a bit about nuclear medicine. You don't know much about it. You might, correct some of the stuff you come out with all the time. Yeah. So that's what I'm doing. I'm doing.
You took it to heart, and you're doing it in a big way.
Yeah. So last couple of weeks I was in Europe, and I did a 1 week course on the medical physics of nuclear medicine at the Royal Marsden. A fantastic course. We'll put the link in the show notes.
Fantastic. Yeah.
So I was there with a whole bunch of medical physicists and a few radiology registrars, a few nuke med registrars, and I sat through hardcore physics. Day one was all about SPECT cameras and advances in SPECT cameras. Day two was about PET-CT. Day three was all about dosimetry. And day four was about theranostic technology. So it was great. Wow. I really enjoyed it. Yeah, the bits I understood.
I don't know what you're talking about right now. That's incredible. It really is.
I know. And thank you to you, because you're running the show at Peter Mac. You've taken over my job as...
Why do you do this again?
Yeah, I know. Well, thank you. Yes, of course I miss Peter Mac, but a lot of the team are here at the Preceptorship.
Absolutely. It's great to have them around. And, one of the amazing things about this conference is that face-to-face conferences are back.
Yeah, it feels like it, doesn't it? There was really a drinks reception, there's a dinner, and it's hybrid as well, of course. And we'll talk a bit about the preceptorship later in the program, because I know a lot of our listeners are interested in PSMA imaging and theranostics, and not everyone can be in Melbourne this week, but the conference is a really good conference for anyone especially interested in theranostics, I think, and that's what we're going to talk about this evening. And this whole preceptorship will be available online, and we'll talk about it a bit later, and we'll put the links in the show notes.
And it couldn't be more timely, really, because really there's been some incredible milestones that theranostics has hit in the last couple of weeks.
Yeah, it is. Yeah, the last couple of weeks, exciting times. Like what?
Oh, well, yesterday was World Theranostics Day. Did you know that?
No. Are you serious?
The World Association of Radiopharmaceuticals and Molecular Therapy, fondly dubbed WARMT, celebrated World Theranostics Day yesterday.
Are you winding me up with that?
No. Our very own Louise Emmett was part of a webinar yesterday talking about, life after vision.
All right, World Theranostics Day.
But that's not the only news.
Yeah.
What else is up?
Yes, we had the FDA approval of Lutetium PSMA. I mean, that was big news for all
amazing.
Prostate cancer.
Yeah, I mean, that's what all this hard work is for.
Yeah, so coming off the back of Vision and Therapy, those trials we've discussed a lot on the podcast, there is now FDA approval and subsequent reimbursement for that, and we're going to talk about that this evening, because that is pretty exciting.
And we've got all the key players back.
Yes, we have. So shall we introduce the guests this evening? So who have we got here in the
all
Sofitel
friends
studio?
of the podcast.
Yeah, go on, shoot. Who's sitting off to you? And by the way, we're on YouTube as usual, as well as Apple and Spotify. So if you're watching on YouTube, you'll see our nice little studio that we've set up in one of the rooms here at the Sofitel.
So right next to me, sharing my Rode microphone, is Professor Louise Emmett, who's just won Paper of the Year, European Urology, at EAU. For the primary trial, yes. Amazing, Louise. Welcome back.
It was a joint effort.
Well, the last time we spoke to you was about the primary trial, and you're off to Amsterdam in a few weeks to grab the award, and it's going to be amazing.
Yep, I think a few of us are going to be there, huh?
Yeah.
At the EAU to grab that award.
Yeah.
You know, it's a great achievement on behalf of all of us.
Yeah, absolutely. And we've got an amazing guest for the preceptorship, Professor Tom Hope. Nuclear medicine physician from UCSF, I mean, completely integral in the trials that led to the FDA approval of gallium PSMA diagnostic imaging back in 2021.
And not joining us on Zoom this time?
No, he's in the flesh.
In person. Tom, welcome.
Maybe someone will see that I'm here and I'll get into trouble. I don't know. Is he not wearing a mask?
Amazing to have you here.
Welcome to Melbourne, Tom. Great to see you.
I'm excited. It's fun to actually be able to get on a plane.
Yeah. And Tom loves long trips on the plane, so I think we can see him here every now and then from now on.
Yeah, well, look, great to have you, and you're here, of course, for the ProsTIC Preceptorship, and we're looking forward to engaging with you. And you were at Peter Mac today and yesterday for tumour board meetings, and it's been really, really great to have you here. So
it's been wonderful.
Yeah, and we want to talk about the work you've done that's led to the approval of PSMA PET-CT for imaging, FDA approval a couple of years ago. We spoke about it on the podcast, and to get your perspective on Theranostics, Lutetium PSMA.
Looking forward to it.
And of course, our very favourite friend of the podcast, Michael Hofman. Does he need an introduction?
Yes.
You
He likes being introduced.
You do the others.
Yes, Professor Michael Hofman. Michael, welcome, nuclear medicine physician extraordinaire at Peter Mac, our close collaborator, who has led many of our most exciting trials in Australia, Pro-PSMA, the initial phase 2 study of Lutetium PSMA published back in 2018, and therapy with your collaborators at ANZUP and others. So, Michael, friend of the podcast, and you're chair of this ProsTIC Preceptorship. So, thank you for joining us this evening.
Welcome back.
Thanks, Renu and Declan. Pleasure to be here.
Good to be back. So, what are we going to talk about?
Well, there's so much to talk about, but I think we should hand it to Michael because he tells a great story.
Yeah, let's go back.
Yeah,
Michael, we wanted to, people are reading about FDA approval, and, we've got these headlines about imaging and agents getting approved, this, that, the other. But for many of the listeners out there, it's all a bit of a blur. So what we wanted to do was ask you to take us back to the start of PSMA theranostics in prostate cancer that led to where we are now with suddenly FDA approval and all that. So how do we go from the starts of that right up to where we are? Because it's quite interesting the way radiopharmaceuticals get developed, both for imaging and for theranostics, and we're really interested in hearing your perspective as a reminder on that. And I'm sure Louise and Tom will chip in with their stories, because that's what you folk do. You enjoy learning about peptides and little evolutions as things goes on. It's very different to the pathways for drug development, I think, in oncology traditionally. So, take us back to the start of PSMA, Lutetium PSMA.
Well, we have to go back a few years, even before I was born. Maybe as far back as 81 years ago, because it is World Theranostics Day, and that actually celebrates the day Dr. Saul Hertz gave the first dose of radioactive iodine to someone with thyroid cancer. And the thyroid gland's the only gland in the body that uses iodine, and he had a brilliant idea at the time. It was in the early days of radioactive substances when cyclotrons that could produce these substances were just being made, and he asked the question, if I can give a radioactive form of iodine, could I treat thyroid cancer, because it's not taken up anywhere else in the body. So that led to radioactive iodine, and we've been curing patients with metastatic thyroid cancer for just over 80 years, which is quite remarkable. These are patients with lung, bone metastases, and with a single dose of iodine, you can cure them. And that's because you get these incredible doses, like 200-300 gray to sites of metastases. And with a 1 millimeter path length, similar to the lutetium we're using nowadays, it's really very targeted. But my foray into this avenue was probably coming to Peter Mac in 2009 and being absorbed in the world of neuroendocrine theranostics, much like Tom Hope, who's a sort of world expert in NET theranostics, and we used lutetium dotatate to image with gallium dotatate neuroendocrine tumours and to treat patients with neuroendocrine tumours. These are rare tumours that arise in the pancreas, small bowel, lung And we see incredible results. And that really fine-tuned my understanding of theranostics and enabled me to see the potential. Probably back in 2010, around 12 years ago, I was at the European Nuclear Medicine meeting in Birmingham. You were probably not too far away at the time. Were you already in Australia?
Well, I certainly wasn't at a nuclear medicine meeting
No,
back
no.
then.
Maybe you were there. I saw my first PSMA images. I think they were, blurry images. That imaging technology wasn't as good, but it struck me that it looked much like dotatate in neuroendocrine tumours. There was super duper uptake in prostate cancer, hardly any background. So since you can see what you can treat, if you switch from a diagnostic to a therapeutic radionuclide, you're going to be onto a really good thing. So these images really struck a chord with me. It was kind of my image of the year. And John Violet, a radiation oncologist at Peter MacCallum and me, got talking about this, and we said, we're going to do a trial of lutetium PSMA in prostate cancer. At the time, there was some very early compassionate access data coming out of Germany showing good results, and we were like, we're going to do this prospectively, properly, because what we learned from net theranostics was all retrospective compassionate access data, and it took kind of 20 years for neuroendocrine theranostics to take off, because in the US, the FDA rightly demands high-level evidence. They want randomized controlled data, and we had learned from that experience. So we, in 2013, started writing this trial protocol, and it was published in Lancet Oncology in 2018 with that famous image of the year showing spectacular responses, and leading on from that, the therapy trial. But we've seen it transition from really a very small, almost backyard exercise. At the time, PSMA 617 was distributed by ABX, a chemical manufacturer in Germany. They used to ship us the PSMA 617. We'd get the lutetium from a number of providers. We'd cook it up in our radiopharmacy. Industry wasn't really interested. Is this thing really going to work? You guys had never heard of theranostics. None of the prostate cancer doctors were really interested in what's that.
We were urologists at that time, not prostate cancer doctors, right?
Yeah. They told me Theranostics. That's like Theranos, that company that's really dodgy. You don't want to get involved in that. I'm like, no, it's completely different. Nothing to do with it. And, that was a very successful 30-patient, 50-patient trial, and really got industry very interested. So a US company that Tom knows very well as well, called Endocyte, purchased the intellectual property rights for this PSMA 617 molecule. We should, let the world know that was really some fundamental work done in Germany. PSMA is not a new target. It's 20 years old, but some German chemists, together with some US collaborators, developed some small molecules rather than antibodies targeting this receptor. And there were two main molecules, PSMA 617, developed in Heidelberg, and PSMA INT, very similar molecule, developed in Munich, and they both sort of were used in patients back as far as 2012, 2013. And I was lucky enough, like you're on sabbatical now, I was on sabbatical in 2016, and I got to spend some time in Germany, both in Munich and in Bad Berka with Richard Boll, and spent that time executing these trials kind of remotely, getting it happening, and it's been an incredible journey.
So ABX was a small company in Germany that had acquired the rights to this thing, and they sold it to Endocyte. Was that like billions of dollars or?
That was a big transaction at the time. Don't quote me, but it was maybe $20 to $80 million.
It was 20.
It was less.
Less.
It's like 10 or 13, wasn't it?
So they bought, but it had a whole bunch of
there was
royalties.
a licensing royalty.
It was 12
payments.
million with royalties.
Yeah, it was very small.
It was about 12
Very
million.
small at the time.
Very significant royalties.
Okay, so that's interesting.
And
then Endocyte have the rights to this thing, which has had some retrospective data, and the pictures look very pretty. But how is it going to get into? That's not the way normally oncology pipelines work. There's phase 1, phase 2, phase 3. I suppose maybe even 10 or 12 million. You might have argued might have seemed like quite a bit of money at the time, really. But so how did it get to? That's what 2015 or 2016.
I think
or
it's
not even that long.
They took a punt. So
Yeah,
what they did was they bought it for 12 million with royalties, and then they said, well, we're going to design a phase 3 trial. We've got all this retrospective evidence, and they went straight into phase 3 trial design. And they're already
company.
For a small company, because they had been doing quite a lot of phase 1 and phase 2 trials, looking at PSMA not theranostic agents, but other PSMA agents. I think they had PSMA ADC before that, so they understood something about it. They took a punt, they did a phase 3 trial, and then they sold onto Novartis before the phase 3 trial read out.
I know that it was such a punt. They did have access to our Peter Mac phase 2 data.
They did.
So Endosight, I think I can disclose, they flew out to Peter Mac, really had a good look at our phase 2 data before they purchased it. And then, Endosight purchased it on the background of all that retrospective data with a very small amount of prospective data, only 30 patients, but it was pretty compelling data. And then the Novartis purchase was how much, Tom?
$2 billion.
Two what?
Two Bs.
Two Bs, wow.
I remember that time, Michael, because the phase 2 data, the Peter Mac paper that read out in 2018 was the first prospective data. But it was only like single centre. These were poor men with very advanced prostate cancer. But at least they'd been treated prospectively. There was a trial, there was a protocol. It got published in a good journal. It was Lancet Oncology. And I remember that. It was about July or August that year, 2018.
Yeah.
And then in about September, October, Tom, as you just said, it was suddenly announced, stock exchange announcement that Novartis have acquired this Endosight company for $2.1 billion US. And in this press release, they said, cited this phase 2 trial of like 30 people, expanded to 50, published in Australia. So is this just me, but didn't it seem like an extraordinary leap to go from a $10 million company to a $2 billion company based on a single center, single site, single arm prospective study? That seems like...
I don't think they bought it. I hate to burst your bubble. They did not purchase it based upon the Peter Mac trial.
Sure, yeah.
So the thing to me that's most amazing about it is the FDA green-lighted going to a phase 3 off of really at that time, which was the retrospective German data, right? That was what was really the thing available and convinced people to move forward. I think, moving forward, they use and leverage the prospective trial data from Peter Mac. But I'm amazed that the FDA accepted retrospective compassionate use trial out of Germany and said you don't need a phase 1 or 2 and just let them go straight to a phase 3.
But I think, Tom, that really... There was a lot of oversight over our Peter Mac phase 2 data that did enable that. There was, I think without that prospective 30-patient trial, it may well be that it's still in the backwaters. Someone needed to do that prospective trial in order for it to explode.
It's interesting. I think one of the disadvantages of what they did going straight to phase 3 is we didn't get information on dose.
Correct.
And that's a real loss. There has been a dose escalation trial, but we have empiric doses that we're using in all these trials, and it almost feels like it's set in stone at the moment. And that's if you could go back and do something again, then having a dose escalation trial that we did in the beginning is something I really would have liked to have seen.
Which again is not normal oncology pathways, is it?
Yeah.
Really? Normally we're used to phase 1. You're finding is it toxic? Phase 2. What's the dose? Phase 3. Is there a benefit? But I suppose, Tom, that's why it's different in radiopharmaceuticals, isn't it? That's why FDA probably had to think a little bit outside the box to say, well, it's a therapy, but it's not going to be like an oncology drug that we normally develop. So it was a little bit of a leap of faith, maybe.
Radioligand therapies just in general are very different from oncologic agents in the sense that, oncologic agents, you usually go up to an MTD, right? It's insane in the radioligand therapy world to be like, let's go to the MTD and dose just under that, right? Because that's not the goal. You're not trying to give the maximum tolerated dose. You're trying to give the maximum effective dose, right? You want to control the disease. So it's a very, I wouldn't run a phase 1 trial where you go until you knock someone's kidneys out, right? But that's what you would do in a regular oncologic therapy, right? You give them until you get dose-limiting toxicities. But I don't think that model is totally applicable in a radioligand therapy environment. I don't want to go to a point where I'm seeing significant toxicity and then we'll dial it back by 20%, right? That doesn't actually work. So I'm not actually sure what a phase 1 trial really should look like. It's a little bit hard to actually fathom how you would do that properly.
Yes, it's really quite difficult. It would take quite a lot of thought, but it would be nice to see one.
Oh, yeah. Well, it would be nice to know, and you take Dotate's a great example, right? We're underdosing in some respects. It's a very effective therapy at the dose we're giving, but we could definitely give a lot more into the right patients that need it. And that's the hard thing. It's not exactly that everyone should get twice the dose. It's just giving the right dose to the right patients. And that's where unit dosing standard scheduling limits us and shackles us, right? It prevents us from being as effective as we could be.
The challenge is, and we learned this from our radiation oncology colleagues, you would know, you give external beam radiation, the side effects are delayed. It doesn't happen a week later. It doesn't happen 2 weeks later. It's not like a drug. You know, the renal toxicity after a single dose of lutetium could be 9, 12 months down the track. So if you're going to do a dose escalation design, are you going to wait a year until you go up to the next dose? You can't do that. So there are these nuances that mandate a different way of thinking about it. Plus, we can also measure dosimetry very accurately. Tom and I, you know, we love dosimetry, but we have a bit of a love-hate relationship, or maybe Louise as well, with dosimetry. But you can measure. You can't do this with a drug. You give docetaxel, how much docetaxel is getting to tumour, you've got no idea, you can't see it. We can measure it very accurately.
So I was going to ask you, Renu, now. So I'm on a sabbatical.
I know you.
Right? I know this shit.
You do know this stuff.
In my course at the Marston, there was a whole day on dosimetry.
Yeah.
So
Did you have an exam as well?
Can I just relapse back into Peppa Pig level of explanation of the way
okay.
nuclear medicine works that I've learned. So unlike in traditional oncology where there's a phase 1 thing, you're finding a dose that's tolerable, phase 2, or that's safe, then phase 2 tolerable, etc. You have this unique ability to measure the dose with dosimetry. So you inject this radioligand, it's intravenous radiation, and using SPECT cameras you can measure the dose. But the thing that appears to me is that you have to measure it not just one time point, you have to have like 3 time points. I've learned about Monte Carlo curves and, iterative blah, blah, blah. But it's a bit of a limitation. But the attraction potentially is that you inject this thing into a patient, you measure the dose that their individual tumour is receiving, and then perhaps you can actually adjust the dose individually for the patient. So unlike traditional oncology where it's done in a mass population thing, this is the dose for this disease. With radioligand therapy, you could measure the dose the patient has received and then adjust the dose potentially for the next patient. But there are limitations, and you folks have all published these beautiful papers on dosimetry, but it requires the patient coming back the next day, 4 hours later, the next day, and a couple of days later. But on this course, we were discussing this topic of what about single time point dosimetry, etc. So the question I have for you on the back of that discussion is it not feasible that you could have a completely personalized approach to the way we give lutetium PSMA to prostate cancer patients to say, you folk have just identified that we didn't get the dose. We didn't do a phase 1. We don't know the dose. But you have an opportunity to say, well, this is the measured dose into my patient. And therefore, in the next cycle, I'm going to increase it to 8.8 gigabecquerels because you've done the dosimetry. Is that feasible, or am I just too enthusiastic after my little foray into the world of nuclear medicine?
I think we should let Tom answer.
Let Tom answer first.
I have a dream, It would be that's where we need to go. It's not going to be tomorrow, and the problem with that model is that it only looks at half of the problem. All right, you have dose, and then you have the tumor sensitivity to dose, right? So every patient doesn't respond the same to 40 gray or 70 gray or 100 gray, right? So there's a tumor-specific intrinsic sensitivity based on genomics, probably all sorts of other stuff.
But our external beam radiation oncology colleagues, they don't adjust based on the tumour genomics. They go, I'm going to give 70 gray. True. They pick a number.
And they cure all prostate cancer, obviously.
Right.
But you need to take that into account. So every disease is different and different stages. So the dose that's effective in a castration-sensitive patient is going to be very different than a castration-resistant patient, and where you're at in your stage of disease will change dramatically. And as has been shown really well by your group, is that you give patients dose, and some patients respond really well to the same dose absorbed in the tumor. And so we need to understand that. But I desperately want to use dosimetry to be more patient-specific in our administered dose. I don't think in America we're never going to be able to do what you do in Australia. Give 8.8 gigs, give 5, give 7. We're going to be stuck at 200 millicuries per pop. The thing we can adjust is number of cycles and the frequency of cycles. That's what we would have control over. But the way our regulatory system works is too hard to be adjusting administered activity in that way, unfortunately.
Well, I mean, luckily we do have some upcoming trials that are in progress right now that is looking at dosimetry as a primary endpoint and looking at this, how much dose we can get into the prostate.
Dosimetry is a primary endpoint?
Right. Could be.
Yeah.
It doesn't
That's
sound
a
like a
novel
surgical
idea.
trial.
Well, it is. Well, yes. Thank you. And for listeners, yes, we're running a trial at Peter Mac that myself and Michael are co-PIs and called Lutectomy, where we're giving lutetium PSMA prior to prostatectomy. And the primary endpoint is dosimetry. We're trying to see what sort of dose can we get into prostate. But I'm on a sabbatical. I've now appreciated that it's difficult to measure dose in if you're only measuring it in a prostate, in a localized cancer, or maybe a lymph node, partial volume, etc. But I still think I still think
I'm surrounded by new
a bit of
things.
knowledge, just a tiny bit. Hey, what were the other things I need to drop in that I learned about? Anyway, but I understand that. But I think we as clinicians working in prostate cancer, if we're embracing this era of radioligand therapy, we, not nuke med physicians, we urologists, nuke med, radiation oncologists, medical oncologists need to understand a bit more about dosimetry, the limitations of it, but the opportunities of dosimetry. Yeah. So I have a dream, Tom. I agree with you. And I think that's probably an area that deserves more support, more research support on how we optimize doses for patients. Is that a fair enough way to kind of round off the idea that we don't have phase 1 studies? It's a different pathway, but we have opportunities to try and optimize doses for our patients who are having lutetium PSMA.
Yeah, I find it interesting because for me, I think the first dose with prostate cancer is really important. You've got very bright PSMA avid disease. When you give lutetium, you've got positive PSMA positive cells right next to PSMA negative cells. And one of the real advantages of lutetium PSMA therapy is you get this cluster bomb effect. So the really bright cells actually help kill off the cells directly adjacent. That's your first dose. And one thing I would say against dosimetry is that you figure it out after the first dose. So you work out what you've just given. So perhaps we actually need to rethink it. How can we do it on the PET? How can we do it on the PET? Can we do dosimetry on the PET so our very first dose is best bang for buck?
And we have some data from therapy that was presented at ASCO GU on our therapy imaging biomarkers, and actually going to deep dive. There's not much science at the preceptorship meeting in the next 2 days. It's mainly education, how we do it. But there is one presentation where you're going to get a sneak preview of extra data from that therapy biomarkers analysis. And what we see, when you do a PSMA PET scan, your SUV, standardized uptake value, how intense the uptake is, there's enormous variation, kind of tenfold. You can have an SUV of 10, which is the lower end of maybe what you would treat. Vision was above liver. Liver has an SUV of about 5 to 7. So you were treating anyone with an SUV above 5 to 7. But last week we treated someone who had SUVs of 330. So that's more than tenfold your vision entry criteria. That means you're getting 10 times the radiation, roughly, compared to an entry-level vision patient. These are enormous differences. And if we wind back 80 years to radioactive iodine, the SUVs we get when we use I-124 and a PET scanner are astounding. We see SUVs as high as 4,000. So there's no wonder that radioactive iodine is so effective in soft tissue metastases. It's not magic. We just get huge doses of radiation with short path length. So we can measure this and it's quantifiable, and then we can predict outcomes.
There was a podcast I was listening to from your colleagues in New South Wales recently, Louise. There's a guy called Dr. Harry Marquis, who is doing his PhD, I think, on using PET to measure SPECT. It was very compelling. I'll put the link in the show notes on a very good podcast called the Nuclear Medicine and Molecular Medicine podcast from Rob Williams and team. But his project is all about using PET to measure it. So I think that's what Michael's referring to. Maybe that's what we should be aiming for. But anyway, we could get really into the depths of dosimetry. But Renu, we've got a few minutes left. There was a few other big things we want to visit with the group.
Yeah, I guess the main message is that we have FDA approval now for the first radioligand therapy in metastatic castration-resistant prostate cancer, and it's strangely labelled Pluvicto.
Can you pronounce it?
Pluvicto.
I haven't practiced enough.
Pluvicto.
That's
Pluvicto. But as well as a gallium PSMA preparation kit.
And I love the name. It's Locomets. I couldn't think of a better name myself. Locomets.
Yeah.
I don't share that love, but
Locomets.
The FDA's wiped clean PSMA 11 from all the labels. We're having to adjust our label to remove PSMA 11, so it's gazotitide. So all of our American literature will be random words you've never heard of.
Wow.
Right?
Is that right?
Yeah, it's disappointing. I mean, why not just call it PSMA 11? It's what everyone calls it, but
Hang on, but
the way
doesn't
we name drugs.
I read the brochure for therapy, and it said you have to have a PSMA 11 scan.
Yeah,
they use
that's
that
going to
word.
go away.
That's going away.
Or what about DCF-PYL?
That's Pylarify, isn't
That's
it?
piflufolostat.
Oh, you're
that's
good.
right. Nice. Well done.
Sorry, what was that?
What was that again? 3 times fast.
Piflufolostat.
Is there an extra F in there?
It's a lot of L's in there. I don't know how they came up with that one.
But if the FDA label for the therapy is saying PSMA, gallium PSMA, does that mean, because DCF-PYL we'll call it, the fluorinated PSMA tracer is FDA approved for detection.
Mm-hmm.
Is it also then approved for selection of patients for lutetium PSMA?
So no, I mean, well, it's interesting, right? So if you read the label of PSMA 617, it says Locomets or an approved PSMA 11 agent. So that would include, so Telix has a kit approved, Illuccix, and then we have our NDAs at UCSF and UCLA. So they would all be considered acceptable by the FDA's perspective. And they clearly specifically did not want DCF-PYL included, right? They went out of their way to exclude DCF-PYL. Now in practice, that will have no value. So it doesn't make it, no insurance company is going to be like, give me your PET report. I want to see what PSMA PET radiopharmaceutical was used to see. It's going to be impossible to police. So it'll end up not being a relevant statement. But it's a reaction. The FDA is reacting to what happened with lutetium dotatate, right? When lutetium dotatate was approved, they said somatostatin receptor positive disease, right? Which is really what the label should have said with PSMA. It should have said PSMA positive disease and just walked away. But what ended up happening in that setting is they all the NETTER-1 trial, you did Artria-scan, which is terrible, right? And everyone ended up using gallium dotatate, and you end up having an issue where patients qualify with very low volume disease with gallium dotatate who would never have been enrolled in the NETTER-1 trial. So there's a sort of a staged migration that the FDA they wanted to limit it to the agent was used for inclusion.
Hang on, so if DCF-PYL is superior to gallium, which is what some people say, then this could be a problem.
Yes, if that untrue statement were true, then it would be a problem. That's true.
And Michael, by the way, I think I just saw a tweet today from European Urology Oncology. You just led a systematic review comparing PSMA 11 with fluorinated PSMA. And in my extensive reading of the tweet that summarised the 3,000 word paper, I think there's not much difference between the two of them. But you're the senior author on that. Are you saying that actually it's superior, or you're saying some people say?
No, I think, as you know, we use both.
Yes.
The fluorinated and gallium in our institution.
They're both very good.
But we're talking DCF-PYL and gallium PSMA 11, and I think they're bioequivalent. I think they're both incredible PET imaging agents. They're both outstanding. But we should differentiate it from PSMA 1007, which is a bit of a different beast. PSMA 1007 has problems. Bone uptake, it's a bit more non-specific. So I think that one is, we shouldn't just say any PSMA. We do need to have some rigour, and we do need to ensure that they are equivalent.
Yeah, but by and large, PSMA 11 and DCF-PYL, that's what we're talking about. So Renu, we've got about another 5 or 8 minutes.
I mean, I think we should now talk about life after, right?
Okay, we started off with let's go back.
Well, I want to ask Louise about the potential competitors. What about PSMA INT?
Yeah. So, I guess Australia, it's very interesting, isn't it? There's been this FDA approval of lutetium PSMA 617, and that's really exciting, and everyone's very excited, but there's silence in Australia for us, correct?
There is a bit of silence until now. Until this podcast. Let your voice be heard, Louise.
It's not the voice of funding and reimbursement.
Silence in Australia. So, what have we been done? We haven't had access except in investigator-initiated trials to lutetium PSMA 617. We haven't been involved in the large Novartis trials in Australia. We haven't had access to them. And we don't have approval pending for lutetium PSMA 617. So lutetium PSMA INT is something that we've all got experience with now, because we are able to buy it from a chemical company in Germany. It is an off-patent agent, and it's available for men in Australia. It's not funded. There's no pharma support for it. But it's definitely something that we're using. And personally, I think it's really equivalent to lutetium PSMA 617 in my experience. What do you think, Michael?
Yeah. it's an interesting agent. You know, actually the very first treatments in Germany were with PSMA INT. It actually slightly predates PSMA 617 use, and you know, we have more experience with 617, but it's INT's had a lot of global use, and certainly speaking to a lot of German collaborators and Louise, we've done a bit of INT at Peter Mac. It certainly seems pretty equivalent, and you've got some dosimetry evidence to guide that. So unlike a conventional drug where, you know, are you going to roll out another phase 3 trial to show that this is equivalent? You know, that's probably what the FDA will require for PSMA INT, and Tom can talk more, but there is some commercialization of INT happening in the States. So that phase 3 data is coming, but you can kind of have some surrogate dosimetry data to just know that the doses to normal organs and tumour is similar, and that data is already available. Yeah. So it's pretty compelling dosimetry data. There's no magic here. We're just delivering radiation to tumour, much like external beam. If you can quantify it and it's the same, you kind of get your answer. Are you going to redo a $2 billion trial just to show that? Seems like a bit of a waste of money.
But the commercialization situation is very different. So we talked earlier about the fact that 617 is now was acquired a few years ago for $2 billion, and it's probably worth a lot more now with FDA approval. But this PSMA INT, is that the patent free type thing that people can mix up and sell ish, or because you've got a couple of different commercial? So, for example, Tom, I hear that the FDA approval will lead to reimbursement costs of about, is it like $50,000-$60,000 US per cycle?
I think it's like 40, but still outrageously
plus
expensive.
A few things, yeah. So PSMA INT, is that a different thing? It's not been acquired for $2 or $4 billion, has it? Is it? So can you please explain that just to listeners?
So PSMA, it's not off patent. It was never patented. It was never patented. So it was developed in Germany.
Yeah, like a lot of that great stuff that came out of Germany.
It's amazing. It's like PSMA 11. It wasn't patented at the time that it was released, and it was available around the world for us to develop into trials. You know, a lot of what Australia's been able to do in terms of developing evidence for PSMA, both with PSMA 11 and also using INT, has been because they're off patent.
Tom can probably comment further on the American context, because in Australia, no one's really commercialising PSMA INT. We all make it in our hospital radiopharmacies. But in the US, there are some groups commercialising INT. Can you tell us about that?
Well, it's sort of surprising. There's 2 phase 3 trials that are in essence identical, using the same drug, run by 2 different pharmaceutical companies. Just sort of, if you think about it, I've never heard of that, right? But if you get approval of it, then they can sell it, and they get 5 years of exclusivity. So with the FDA, if you get a drug approved, you get 5 years of exclusivity, and you know you can make significant revenue, and you sort of know it's going to work, right? It's bioequivalent in essence. So it's a somewhat low risk straight to go into a phase 3 trial. It's not, the difference in the US is we can't. We could label Lutetium PSMA INT in-house if we wanted to, but you can't charge insurance companies for that.
I've got a question. Now that we have the vision data, and you say it's bioequivalent, how are you going to repeat that study? Isn't that unethical? Would you want to be in the non-lutetium arm? How would you run a trial like that?
So that's true. That's why it's not a vision. So there's 3 trials that are identical, right?
Isn't it compared to docetaxel, isn't it?
No. They're in essence all pre-chemo, post-first-line ARRT. So in essence, it's PSMA4, SPLASH, and ECLIPSE are all this. There's slight variations in them, but they're all, you progress Avienza, MCRPC, you do a switch, right? Now, whether or not that's ethical is a different discussion.
So what's in the control arm?
It's second-line Avienza.
We need to get back Vinay Prasad. What would he say about that?
I know what I say about it, and I'm very involved in one of those 3 trials, and I don't like it. The problem is, and this is where, you know, I think it's a really good lesson to learn. Why did the US run vision, and why did Australia run therapy, right? You guys wanted to ask the right question, right? What's clinically relevant? What's important? America wanted to get a drug approved, and not America, but the American market. The companies running, you know, vision was designed to get approval from the FDA. That was the only goal of that trial. The trial was not designed to ask the most academically interesting question, right? And so ECLIPSE and SPLASH and PSMA4, those are trials. If the FDA is going to let you run that trial, you're going to run it, right? There is no reason as a pharmaceutical company to be like, you know what, I need a better control arm, right? I want to make sure that my drug really beats out a really competitive control arm. There's no incentive, right? So the issue here is not the company, it's the FDA. Because if the FDA allows, the moment the FDA allowed the first company to run that trial, every other company changed their trial to look exactly the same. Because, if you were to run a trial comparing something to cabazitaxel, it's going to be hard to win. And the FDA does not approve drugs with you have to have OS benefit, and so you got to have a really significant win over your comparator. So to me, it's the issue of what the FDA is expecting, and they don't change the goalpost based on what the comparator arm is, right? Like, I think it's way more clinically relevant to do a docetaxel versus PSMA, whatever you're doing post Abienza. But as a company, do you want to take that risk? It might not actually have RPFS benefit or OS benefit, right? But it's clinically relevant. It helped me know what to do in a patient, right? Because that's what I want to know. But there's no way a company is going to put $40-70 million on the line when the FDA is going to let you run a trial. It's also weird in the U.S. Over 50% of patients who get Abienza first line switch off trial, like standard of care. Switch is actually commonly done, which is the argument these companies make to say, hey, this is clinical evidence, this is what we do.
It's standard of care.
It's standard of care. And it actually is, oddly enough, even though we all have our issues with it. The CARD trial and things like that suggest it's not the right way to go.
As ever with these trials, it's often the control arm that's the interesting bit. And
right.
we did a Vinay Prasad on, who I know divides opinions, but I think he does very often poke very good holes in industry-supported trials, and he's often got his tentacles into the control arm. Have you rigged the control arm? And is that really standard of care? But, it is what it is. We are where we are, and it's very different here, as we just pointed out. It doesn't really matter to us what the FDA approves or not. We're really thinking, what can we afford for our patients, and what's clinically relevant? What is an appropriate comparator? And it's a little bit more purist. But I think it's interesting because unlike other traditional drugs where you're waiting for something to go off patent 10 years later, what you have in the radiopharmaceutical industry is people are tweaking little things, and now you have 617, a multi-billion-dollar acquisition, but the Germans have now tweaked a little thing, PSMA INT, imaging and theranostics, and that's available. And now you have a couple of different companies trying to commercialize it, and then many others who we can just buy it off the shelf here and cook it up. So it's very different to normal oncology. But I'm encouraged, Louise, to hear you say, well, look, by and large, these things are probably relatively equivalent, which is why you are leading a group that are seeking reimbursement in Australia for PSMA INT for metastatic CRPC, because Novartis have, I understand, shown no interest in taking it through a reimbursement pathway in Australia. I think, is that right?
Well, I think at the moment they haven't shown interest. It may be that they will in the future. And I guess, the question is, how long do we wait? Our patients have metastatic prostate cancer and they're symptomatic now. And we need to fight for them.
Well, it's almost a bit worse, Renu, because the frustration, you've run a trial here a few years ago, you see benefit, does it compare, and then the FDA approval, well, hang on, and we've been using it, we generated evidence 5 years ago.
Still need to make it accessible to patients.
Yeah, exactly. And then the big industry are saying, well, look at, we're going to get $50,000-$60,000 a cycle in the US. Yeah. I'm not blaming everyone, that's just the system over there. But meanwhile, have we seen an application through funding organizations in Australia?
Exactly.
No, we haven't even seen an application. So for us, it's almost, I'm a urologist. I don't have any skin in this game, but for our patients, it's frustrating if you have generated data, you see successful trials, and then in your own country there isn't any attempt to seek reimbursement, which is why Louise and co seeking reimbursement through the agent that we haven't tested in a phase 2 trial here or phase 3. But we know is likely to have the same benefit, is very, very welcome, because at least our patients might access this in 2022 or 2023.
But so I think the Australian market's very different. So we've got experience with radium-223 in Australia, where it was not reimbursed due to really all these complex issues. With a national health service and more equitable access, our government just will not cough up that sort of money if Novartis are going to put together an application with 60,000 Australian dollars, which is roughly the equivalent of the US dollar after conversion. The Australian government's not going to fund that. They're going to go cabazitaxels, your comparator, cabazitaxels, 1/5 of the cost. You need to show cost effectiveness. Yes, there's less side effects, but we want cost effectiveness. So it's going to be very hard to get a price point like that funded in Australia, and Novartis probably know that. Novartis will seek reimbursement of this product in Australia. I'm sure they will. They may already be doing it. Like we don't know what Novartis are doing, so we can't pretend to really know. So they will do that, but they're probably mindful that the Australian government will want a lower price point than what's happening in the US. And the tradition, even with Abienza and other drugs, is we're pretty late in the market in Australia, aren't they? When it's reimbursed for that reason, they'll go after the overseas US market, European market, then come to Australia as a third tier. That's just the way our health service runs.
Oh, well, look, Enzamet is a good example. The very successful OS benefit trial with enzalutamide, published in New England Journal many, many years ago. Huge trial led by Enza. I don't think was there even an attempt to get reimbursement here. So no, here we are. We, more than 1,000-person study, most patients in Australia, many patients benefited, but here we are, multiple years later with not reimbursement.
But we do have we do have PSMA 617 available today at Peter Mac for men, and they're not paying for it. It's all funded through a bit of a complex funding model, but it is available. So there will be some places where the Novartis product is available.
Yeah, totally agree. And Novartis have been great partners in enabling all these studies as well, of course, and they've done a fantastic job on it. Yeah. To say. Renu, are we nearly done? What else? I mean, it's fantastic.
we could talk about this forever, right?
I'm sure the audience are enjoying this, and we will hopefully run another couple of podcasts from ProsTIC this week. Yeah, and it is a great conference.
It's a great conference. Lots of highlights coming up, and hopefully you'll be able to showcase a bit more.
Exactly, and if people want to just Google ProsTIC Preceptorship, you'll see details of how to find this fantastic online experience that's about to kick off here in Melbourne over the next couple of days. So that's about it. Tom, welcome to Melbourne. You're settling in well. The jet lag's okay. You'll be able to speak at the meeting in the next couple of days.
You might have to wake me up, but I'll be there.
Yeah,
I'm in the same building.
He doesn't drink coffee, I learned. That's scary.
Oh, really?
I've never had a cup of coffee in my life.
Oh, come on. He's got a couple of days
way.
to settle in, hasn't he?
I'm normal, right?
No.
Fantastic. And Louise, great to have you down from Sydney again, and congratulations on Primary getting Best Scientific Paper of the Year, European Urology. Love that.
Really,
Thank
very
you.
cool.
Great to be here.
And Primary 2 has just kicked off, so that's very exciting as well. We're involved. We're already recruiting patients into Primary 2, PSMA and early detection, which is very exciting. So great, Renu. Nice to be back in business again.
Absolutely. It's great.
Fantastic. That's all we got time for this evening. We'll talk to you again soon. Thank you. Bye-bye.